Thank you.
Hi, everyone. I'm Hannes Neubauer. I'm a PhD student in the lab of Marco Gallardini. And yeah, today I want to present PanFeed, which is a command line tool, which I developed together with Marco, and with which we want to improve the interpretability of bacterial genome-wide association studies using gene cluster-centric k-mers. And before I get into the program itself, I want to give you a brief introduction to some of the concepts. And as you might have heard over during this week a few times, probably bacterial whole genome sequencing is becoming more readily available, and especially for clinical isolates. And so data sets are getting bigger and bigger. And for these clinical isolates, we also have usually phenotypic information, for example, such as resistance to antibiotics or susceptibility to antibiotics. And with these larger data sets, that is especially interesting for GWAS or genome-wide association studies, because we get to identify more rare variants. And how GWAS works is basically, for example, in this example, we have E. coli strains, one with Fimbriae, one without, and then we want to have a look at what genetic variants determine the phenotype of these strains. Another concept that is quite interesting for bacterial genomics is pangenomes. Pangenome, of course, being the concept of like containing the entire genetic variation of a species, while also maintaining the information for each individual isolate. And they usually consist of a core genome and an accessory genome. The core genome containing genes that are present in all strains or most of the strains, and the accessory genome containing genes that are present in many, some, or even just one strain. And with that, yeah, we want to use this concept, especially for GWAS, because it lets us circumvent the reference genome, which doesn't really capture the whole heterogeneity of bacteria. And so the usual workflow, and I will be comparing our program to Unity Counter quite a bit, which is a program that would be used to get presence-absence patterns of genetic variants, which was used before our program in GWAS quite a bit. And so it would start by getting the genomes of our isolates, cutting them into k-mers, which are nucleotide sequences of length k, assembling them into de Bruijn graphs, which look something like this. And then these de Bruijn graphs can be further compacted to unitics, which takes k-mers in a unilateral path on the de Bruijn graph and compacts them into these unitics. And that just reduces the redundancy of our k-mers. And then you get this presence-absence pattern for each unitic. And in contrast to that, we have our workflow, which first of all uses Panaroo, which is a program that creates a pangenome on a gene cluster basis. It starts out with the annotated genomes, and then takes the genes of those genomes and groups them into gene clusters of similar genes. And we then can take this, the resulting gene cluster presence-absence matrix. And then PanFeed comes into play. This is our command line tool. And it takes each gene cluster, and for each gene cluster, it cuts the genes that are present in that cluster into k-mers. And so the k-mers we produce are specific to each cluster. So we can have a k-mer that occurs in two clusters, and in each cluster it can have a different presence-absence pattern. And first of all, this avoids the use of a reference genome, and it lets us also look at the k-mers in a local context. Also, we record the positional information of the k-mers, which makes the interpretation of the results a bit easier for the GWAS. And in a bit more detail, the workflow would be we start out with our genomes, annotate them using, for example, Proca, input the annotated genomes into Panaru, get our gene cluster presence-absence matrix. That and the annotated genomes can be input into PanFeed, and PanFeed then produces 3 files. One containing the k-mers and the locus information, one containing the presence-absence patterns and their hashes, and then one file that is basically a conversion matrix that correlates these 2 files. The presence-absence patterns can then be input into PySeer, together with additional information to do the actual GWAS, and then we get our associations, input that into convenience scripts of PanFeed, which then correlates the associations to the k-mers and their positional information, and then we get this final table. Now, because we noticed that this k-mer file can get quite large, we came up with a 2-pass approach, which basically runs PanFeed twice. The first time, we skip the recording of the locus information and the k-mers, and just produce this these presence-absence patterns, input that into PySeer, get the associations, input that into the convenience scripts of PanFeed, and from that we can get the names of the clusters that are significantly associated to the phenotype in question, and also the clusters that contain k-mers that are significantly associated to the phenotype in question. That can be input into PanFeed as a list, and then we only log or record the k-mers and the positional information of that k-mers for these specific clusters. That again can be input into convenience scripts, and then we get our final table with significantly associated k-mers and their locus information, only for those clusters that are actually of interest to us. And some use of PanFeed that I want to highlight is the investigation of promoter regions in the context of their genes that are upstream of the promoters. And for that, we used a data set of 1,556 Neisseria meningitidis isolates. And for these strains, there was also the phenotypic information of whether they caused invasive meningococcal disease in their hosts or were just carried by their host. And so for this visualization, let's start out on the x-axis, which is the relative nucleotide position relative to the gene start. And then on the y-axis is a subset of 16 strains, which is divided by the horizontal black line, which divides them into phenotype positive strains on top, so strains that cause invasive meningococcal disease, and phenotype negative strains on the bottom. And this is just the visualization of the nucleotide sequence based on color, basically. And then the plot on the right is a combination of the nucleotide sequence and also the negative log 10 of our association p-values as opacity, which basically means that the stronger the color for these nucleotide positions, the higher the association p-value for that specific position. And in previous studies, in this specific gene, they found a single nucleotide polymorphism at position -7 in the promoter region. that was significantly associated to entry, to the causing invasive meningococcal disease. And actually, we also found that using pan-feed. Now, to basically compare pan-feed and unity counter for just to validate that we also get like useful results, we used a data set of 912 E. coli isolates, for which the portal of entry into the host was given as a phenotype. And similar to the plots before, we can see the nucleotide position relative to the gene start on the x-axis, and then our strains, this time the full 912 strains on the y-axis, divided into phenotype positive and phenotype negative. And this time the colors represent the association p-value, meaning a brighter color is basically representing a lower association p-value or a more sound association. And when comparing the unitics of unity counter and the k-mers of pan-feed, you can see that with k-mers from pan-feed, we get a bit of a clearer picture, which is less convoluted. And pan-feed also lets you get the association for genes in general. So whether the absence or presence of a gene in total is responsible for a specific phenotype. And so this is the gene cluster group 5515, which was just the name given to PUBG2 by Panaru. And PUBG2 has been identified before being associated to pyelonephritis and the entry through the urinary tract in hosts. And something that is also mentionable is that unitics from this group 5515 or PUBG2 have also been found by us in 3 other gene clusters, which were truncated or low frequency, and therefore likely false positives. Something that I also want to highlight is the work of my colleague Bamu Fufodamaris, and she's investigating genetic variants in promoter regions being associated to an upregulation or downregulation of the gene expression. And for that, she's using a data set of 117 E. coli strains, for which the gene expression was compared to E. coli K12. And the data set was produced by Nossos Typers, Matilda Zitek, and Marco, my supervisor. And so when, first of all, looking at the association, we can see this specific variant is associated to an upregulation or to a change in gene expression. And when we then correlate that to the expression values, you can see that if the variant is present, we have an expression level that is closer to E. coli K12, and without the variant, the expression value drops below that of K12. For this example, also, when the variant is present, we see a drop in expression level, while the absence of that variant maintains the same expression level compared to E. coli K12. And here it's a bit different. We see an increase in expression for strains carrying that variant, and strains not carrying that variant have a decrease in expression compared to K12. Sorry. Now, for the comparison of resource management and running time, we used FSM Lite and Unity Counter to compare pan-feed against. And so for larger data sets on one core, pan-feed is actually slowest, but for the memory footprint, we managed to be more closely to unity counter, while FSM Lite does its thing. On 4 cores, we managed to reduce the running time quite a bit, as a trade-off, of course, for the memory footprint. And then when using this 2-pass approach that I was talking about earlier, we managed to drop both the memory footprint and the running time quite a bit. And also the camera file is reduced in size from 0.5 terabyte to a few megabytes. And so in summary, yeah, we managed to show that pan-feed largely overlaps in its results with unity counter, while also having the benefit of checking whether a gene in itself is associated and not only k-mers within that gene. And also, with pan-feed, it is a bit more easier to examine these promoter regions in their local context of the gene that they govern, basically. And in general, not using a reference genome that is not able to capture the genetic heterogeneity of the isolates that we want to investigate is also quite a big benefit. Now, there's a preprint out on bioRxiv, so go ahead and read that. And pan-feed is available on GitHub with Conda and PyPy. And with that, I want to thank the whole Galladini group for the support, and of course, Marco, with whom I developed this program together. And then also thank you for your attention. And if there are questions, I am happy to take them.
So do we have questions for the audience? And if you're within running distance, I'll bring you the mic. I have many questions. So GWAS is a very sort of fine-grained analysis, and commensal versus pathogenic is a very coarse. Is that a mismatch? And do you have, if so, do you have ideas about sort of how you get finer grained phenotypic observations?
Sorry, could you, I didn't quite get the.
Okay, so you have, it's pathogenic or it's commensal, very crude, versus this SNP versus that SNP. Is the sort of fine-grained nature of SNPs, do you have enough information with just 2 possible phenotypic states to make good use of the information in the SNPs? And if not, do you sort of see in the future a way of refining your phenotypic information?
I mean, so for phenotypic information, often it's a binary phenotype, but we can also have a continuous phenotype, for example, in MICs, which isn't just susceptibility or resistance, but we can have these fine-grained MICs. And with that, we get a bit of a clearer picture then, I would say.
Have you tried that out? Do you have some data with that more continuous phenotype information?
No, actually, for pan-feed, we haven't tried that out yet. Haven't done that on a data set with continuous phenotype. But yeah, we'd be happy to.
You should. I encourage you to do so.
Yeah, yeah.
What about statistical power? How many strains do you need in order to be able to be confident in the associations that you beat?
So for rare variants, it's quite difficult. So in a previous study done by Marco on that 912 strain data set, he did find the association to PepG2, for example. But he then used, there's a tool you can use to look at the statistical power of your GWAS analysis. And I think for rare variants, he mentioned that we would have to like have data sets of maybe 10,000 strains then. But I think that's definitely possible in the future to have these data sets.
With good assemblies.
Yeah, yeah.
Do we have questions from Juno? Anybody? Do I get to ask more questions, or has somebody woken up in the audience? I have one more, which is you say you can do association with presence, absence, and with SNPs.
Hmm.
Can you put them on top of each other? And do you have any sense of sort of what matters more? Do you learn more by saying it matters if this gene is there, or does it learn more about, I have this SNP, for example, in a promoter region?
So we are not, basically we are not considering like SNPs. It's just the presence-absence patterns of the k-mers that we are looking at. And then from the visualization, we can then see, okay, there's a SNP, and that is associated then because it lies in the.
So you're not looking at phenotype associated with gene absence versus phenotype associated with a SNP?
No, we are just looking at the presence-absence pattern of either the k-mer.
I misunderstood you.
Oh, sorry.
Okay, thank you.